Hello everyone. Now a bit short introduction. What I am gonna present today. First of all, I wanted to say that this recording will probably be uploaded to YouTube, and if you cannot attend or something, feel free to watch the recording on YouTube, and you can watch it also at playback speed 2 that I really like doing, and I sometimes maybe watch recording twice, between different periods of time to remember better if the topic is interesting. And although I couldn't attend this webinar, I would say it was really interesting, and I learned a lot about gene therapy, and if you haven't checked, so please be welcome to check it. Now, regarding other our journal clubs, I thought that, yeah, as we just talked, that everyone is busy with their schedules, exams are coming. So I thought maybe we could do something that would be related to our exams. Like, at least I can talk about my exams now. So I thought to still run journal clubs every week from now. And this would include only 4 journal clubs till April, till I think holidays. So I thought this one to be about epigenetics, another about collagen gene mutations, and then about oncogenes and CRISPR-Cas editing system. But now this one would be about its applications. So most of, like, 3 of them are from this module, and collagen gene mutations from this module. I don't know if you're interested or not. If you want me to change the topic, we can choose from these topics. So let me know, write to me, we can deal this issue and can change topics if you are willing to. Then today I would like to talk about chronogenetics and genetics of sleep. And there is need to note that these two are not the same. Chronogenetics is a much broader term. It encompasses genetic regulation of gene expression in all cells, whereas genetics of sleep refers only to sleep. So now, like nowadays, like in every day, we can feel the timetable, either of bus, either on the streets when we see traffic. Traffic is fully packed when we know that people are returning from works. At school, children are coming at a particular one time at the classroom. But have you ever thought if cells have a timetable? And actually the answer is that yes, most of the cells have that timetable. They express genes at a particular time of the day. And another point I wanted to touch is that in treatment, we try to apply the treatment more specifically. So either by gene therapy that targets a specific gene, thus minimizing side effects on the body, or we want to use personalized medicine. And I thought another way to make the treatment more precise is to give the treatment at a particular time of the day, so that a particular cell type or organ would be affected more by that drug, because that cell would be simply more active at a particular time of the day. And example of those cells could be control of glucose metabolism or cholesterol metabolism. And the two, it could be linked with hypertension, and this with hypercholesterolemia. And these two contribute a lot to cardiovascular diseases, as you know. Now, regarding chronopathologies, I'm not going to go into detail into this, but I found a very interesting paper. Sorry, I forgot to put the name, the title of a paper, but this diagram is taken from that paper, and it simply described which not diseases, but how do you say that, attacks, those symptoms of the diseases are mostly apparent, like at which time of the day. So is this morning, early afternoon, late afternoon, or during the sleep? So for example, stroke is more likely to occur during late afternoon, whereas, for example, angina pectoris is probably most painful in the mornings. So, and I tried then to make circles on those events related to cardiovascular diseases, and it turned out that all of these are related to the heart, like congestive heart failure, early repolarization syndrome, agotonic atrial fibrillation, etc. So that chronopharmacology, chronopharmaceutics would be potential treatment of cardiovascular diseases, improvement of that treatment. And in Google Scholar, when you enter chronopharmacology, you can see quite recent papers on that, and two of them are talking about cardiovascular diseases. One is talking about neurological diseases, and another is talking about ingestion time differences, and also about hypertension. So this paper in 2021 said that there are two critical points of relevance to the practice of clinical chronobiology and chronotherapeutics. So first, that I think I interpret it is that it's hard to decide the time of the day when the chronopharmaceutic should be taken by a person, because every person has its own biological time determined by genetics. So if a doctor says to a person, you should take the drug at 8 a.m., this would be the start of the biological clock of one person, like a circadian rhythm, but not the very start of another person who is maybe, night owl and who wakes up at 12 a.m. So it's hard to, maybe then a patient should know their biological rhythms well, and it's very hard to say for a patient then that chronotherapeutic to give. And another critical point to make is that representative biomarkers of the circadian time structure, the most convenient ones being the bed and awakening times of the 24-hour sleep cycle, enable synchronization of pharmacotherapy to circadian rhythms to optimize effects and outcomes. So now you probably know that there are 3 rhythms, and I gave examples of them at our organism level. So these are ultradian, infradian, and circadian. Ultradian rhythms are more frequent than 24-hour cycles. So it's for examples of that are heartbeat, respiration, blinking, and appetite that you experience like approximately 3 to 5 probably times per day. When infradian rhythm, infra from Latin meaning below, is less frequent than normal 45-hour period, and examples of that are menstruation, migration, and hibernation. And We can guess third one. So third one is circadian rhythm. And these are rhythms that are physical, mental, and behavioral changes that follow a 24-hour cycle. And what's the purpose of those cycles? Why did they evolve? Probably to predict and adapt to periodic changes in organisms' environment. And these rhythms provide a selective advantage, enabling anticipation and exploitation of predictable changes. Now, could you think of any hormone secretion under this control, this rhythm control? Anybody? So you probably see here cortisol and here melatonin, but there are actually another hormone, that is growth hormone, also secreted in circadian rhythm. And you see cortisol here. I found these descriptions of the curves very interesting because they could explain why, for example, you couldn't sleep. Like some people, energy crashes and burns around mid-mornings, and some people who, for example, have intensive focus on schedules during the day. For example, I thought about students, their cortisol level is very high throughout the day, even in the evening, and this is why they could not fall asleep. And then working out at night also could cause increased levels during the evenings. And so circadian rhythms at organ levels, their secretion of various organs, and circadian rhythms at a cellular level are, for example, that cells decide when to divide based on their internal clocks. So now actually what I'm gonna talk further is about Nobel Prize in 2007, I guess. We will check that. And Chronogenetics, and then a few publications related to that. 2017, okay, it's very recent, quite recent. So by Jeffrey Hall, Michael Rosbach, and Michael Young. So what I'm gonna talk about is actually from Nobel.org website. So I will simply explain what we discovered, and these are 3 genes: period, timeless, and double time. And their products are proteins per, TIM, and kinase CKIY. Now a bit about definition of circadian clock. So, you know, circadian rhythm is more like a light and dark cycle, whereas circadian clock refers to molecules in the cell that cause that, for example, cortisol or secretion or growth hormone secretion to undergo circadian rhythm. And circadian clock coordinates internal time with external environment, and is a major regulatory factor for nearly all physiological activities, and its disorder has severe consequences on human health. So these 3 scientists, Nobel Prize winners, they found that mutations in an unknown gene disrupted the circadian clock of Drosophila, and we named that gene period. Then we wanted to answer how could this gene influence the circadian rhythm. And they found out that per protein accumulated during the night and was degraded during the day. And then the question was arose, how could that per protein levels oscillate over 24-hour cycle? And as you see, during the night, there is actually no transcription of that protein. Then when night appears, the transcription starts, and we have a lot of per protein. And when the protein, when it accumulates here, it enters the nucleus. And here I actually found a video of that one of the Nobel Prize winners talking, and I want to show a few minutes. This one, Michael Rosbash. So he simply explained how that period gene accumulates, but I think I cannot actually make it work. Okay, it doesn't work.
There's usually a problem with videos in presentations in Zoom.
Really?
Yeah, like it happens all the time with our lectures, sadly.
Okay. So I have to explain it, but so the thing is that there is negative control of that per protein. When it accumulates in cytoplasm, it enters the nucleus and then acts on its gene to inhibit that gene transcription. So it's inhibitory negative feedback loop. And then it inhibits, there is no transcription, and when light appears, these existing per proteins in the nucleus are degraded, and we get simply, when the light shines, we have again a bit of transcription of that per protein, and when we have no light, we have accumulation of that per protein that again goes into the nucleus, and during the night inhibits the transcription of that per protein. So that Nobel Prize laureate simply told that they had produced a lot of Drosophila with mutations. That, for example, if Drosophila had this mutation, this was advanced sleep phase syndrome. Like advanced sleep phase syndrome in humans, like a model to study, like model organism with this mutation. When, for example, we found that Drosophila had this gene, and mutation in this locus causes sleep duration to be longer. In this region, causes sleep to be shorter, and in this region, this results in stop codon expression. Yeah, so I have talked about it. When proteins not simply go to the nucleus, but they are transported somehow. So this Nobel Prize laureate discovered timeless gene that encoded TIM protein, and when that TIM protein bind per protein, complex of the two proteins could enter the nucleus, and there we blocked period gene activity, and thus we didn't have any inhibitory feedback loop, and period gene could be expressed even more. Now third gene is double time gene that encodes for kinase, and that kinase phosphorylates per protein. Then the per protein is recruited by E3 ligase, and we have degradation in the proteasome of the per protein, and this delays the nuclear entry of the per-TIM complex. So you see here that kinase binding to per protein, and here you see the kinase phosphorylating per and degradation of the protein. And we had insight into how an oscillation is adjusted to more closely match a 24-hour cycle. So these three genes are most important probably because their discovery led to Nobel Prize. And now I would like to talk more about chronogenetics, and so I read this chapter of the book called Chronopharmaceutics by B. Botti and Yuan. So here important terms would be suprachiasmatic nucleus, that is a part of hypothalamus. So you see here hypothalamus, and suprachiasmatic nucleus is that very small thing just above the optic chiasm. Here is pituitary gland. Then this nucleus coordinates subservient endogenous peripheral biological clocks of cells, tissues, organs, and systems through modulation of constitutional clock genes. And I thought this term is very, very, very interesting, because have you ever heard about endogenous peripheral biological clock? When you say about biological clock, you probably would think first about suprachiasmatic nucleus, but there are also secondary smaller clocks in every cells in your body. And so these clocks involve genes such as BMAL1. I'm going to explain what it stands for when clock, pair, and SEER. The product of these clock genes cyclically activate and suppress numerous non-clock genes, giving rise to circadian rhythms of biochemical pathways and most physiological, neural, endocrine, and other processes and functions, and that collectively constitute the so-called circadian time structure. So here we have clock control genes, and that are expressed in every organ. So even though we think about that circadian clock, like day-night controlled clock, actually it's not actually the light during the day does not have to go directly into the cell for it to feel that it's the day. So 5 to 10% of the transcriptome in tissues is under circadian regulation. And we have more of these genes in the liver and suprachiasmatic nucleus. And for example, aorta and the heart have fewer clock control genes. And these genes regulate metabolism, transcription, translation, protein turnover, immune response, cell death, vesicle traffic, ion transport, and molecular rhythms in a highly tissue-specific manner. Then this means that the level of circadian regulation rather than the level of gene transcription is relevant in tissues, since up to 50% of cycling transcripts in one tissue are expressed constitutively in another tissue. E-box is a very important element in promoter of circadian clock genes, and we are present in many of those clock genes, and we are conserved in the human, mouse, rat, and bovine genes. Now, regulation of those genes can be either direct or indirect. So if regulation is direct, then it's regulated by a heterodimer composed of BML2 and clock through two E-box response elements located in the proximal promoter. And direct regulation of clock outputs may not be the most prevalent mechanism, actually. Direct mechanism of clock gene regulation, examples of them could be in the gastrointestinal smooth muscle. So for example, prokinetisin 2 is a potent stimulator of the contraction of the gastrointestinal smooth muscle. Then antidiuretic hormone gene has EBOX promoter. And paraventricular nuclei and supraoptic nuclei of the hypothalamus produce that antidiuretic hormone. Then, as we talked that early morning about myocardial infarction and other cardiovascular diseases. So this infarction is more likely to occur during early morning, and it's mostly because of plasminogen activator inhibitor 1 peaks in the morning. So maybe if we could target that clock gene, maybe with gene therapy, we could reduce the risk of people with myocardial infarction of dying in early mornings. Then glucose metabolism is definitely under the regulation of clock genes, but I was not sure if it's direct or indirect. I didn't check this, so not sure if it's direct. Then indirect regulation of clock outputs. Examples of proteins controlled indirectly would be steroid 15 alpha hydroxylase, cholesterol 7 alpha hydroxylase. And this is a rate limiting step enzyme. So imagine without circadian clock, circadian rhythm, if we would not be responsive for that, we wouldn't have this enzyme, and therefore cholesterol could not be converted into bile acids. Then this indirect control also ensures low oscillation of par-B-zip factors in the brain, so that the brain is protected from vitamin B6 deficiency induced epilepsies. And DEC1 gene controlled under indirect regulation of clock outputs ensures normal immune function. So how do those cells inside our bodies who cannot sense light, how can they know when it's the day or the night? So there is a suprachiasmatic nucleosympathetic nervous system link, and paraventricular nucleus receives a continuous glutamatergic input that is shown in pink. So here we see that nucleus, and those neurons provide glutamatergic, so excitatory input into the nucleus. And we have rhythmic inhibitory input from GABAergic neurons that are shown in purple. And also we have rhythmic activation of a sympathetic input. That is shown in green and goes to the liver. So via intermediolateral column, that is somewhere there, that sympathetic input reaches the liver. So, you know, this nucleus reacts to light, and basically the signal is transmitted through the spinal cord to the liver, and plasma glucose concentration is regulated by the liver. And also orexin neurons also play a role there. Then could you, maybe somebody knows how many genes does a Drosophila have? Somebody wanna guess? So Drosophila has about 134 genes, and 7 genes are actually regulated by clock in the fly, and more transcription factors under the control in the suprachiasmatic nucleus are being discovered, and they are not primarily involved in the circadian clock mechanism. We have other functions. For example, albumin D element binding protein displays a robust circadian expression pattern at the mRNA and protein levels in the liver suprachiasmatic nucleus. Then we have this family of genes, and it's basic helix loop helix family genes that code that examples are DEC1, 2, and these products of these proteins can bind to DNA. To be more particular to E-box elements, and we repress transcription of clog BMAL1 mediated transcription. And promoters of these genes contain E-box elements that are regulated by the core clock transcription factors. DEC1 and 2 have functions shown there, and clock genes actually are relevant because they determine physiological output, and these two are clock genes, right? And how do we know? So DEC1 in mice liver regulates circadian clock outputs, and that DBP protein, the element binding protein, acts antagonistically to DEC1 protein, and they tune the oscillation of physiological outputs. For example, null mutation in DEC1 causes no locomotor rhythm. So the mice without DEC1 protein cannot move. So these are the main clock genes operated in the suprachiasmatic nucleus. And as you remember, we talked about this pair that is very important. And now you see here cryptochrome protein, and that is a blue light photoreceptor. And mutation in that protein causes arrhythmicity in laboratory animals in constant darkness. And these proteins are expressed in numerous peripheral tissues. Cryptochrome genes associate with PER and repress clog BML1 transcriptional activity. Here you see that. Now, BML1 is one of the most important clock genes, and it stands for brain and muscle and T-like 1 protein, and this stands for aryl hydrocarbon receptor nuclear translocator. And these two proteins that you see are quite important because they bind to E-box, and then transcription of clock genes CRE, PER, and this one starts. So we are transcription factors, and we act after we are heterodimerized, and so then we transactivate clock control genes. And here you could relate to the same diagram that I showed just before. Is there per protein? So in the future, we should better understand the pathways that regulate clock-controlled gene transcription. And already more and more physiological processes are found to be under circadian control. And also, a circadian component of various diseases could be more investigated and been targeted by drugs or other therapies. So these diseases are cancer, sleep disorders, metabolic diseases, and cardiovascular diseases. Then we should identify more clock-controlled genes that are related with particular disorders and understand output pathways. Also, it would be really helpful if we understood biosynthetic pathways that are controlled under circadian rhythm, because these enzymes are rate-limiting enzymes. Then clock control genes are relevant for melatonin and cholesterol metabolism, and this also are quite important pathways that could be targeted. When we could identify how many clock control genes are there and their specific functions and tissue distribution. And also we can express level of thousands of genes analyzed simultaneously. And thus, this is a very quick way to study those genes. And the method that this can be carried out is actually DNA microarrays that identify cycling transcripts, and thus we can find extent and functions of clock control genes quicker than many years ago. So mutations in mammals of clock genes are known to cause reduced fertility, impaired cue and contextual memory, increase the risk of cancer, abnormal bone function, and even increase alcohol intake. And now last part is a review of 5 publications and mechanisms that that questions you could have in mind is what is the mechanism and what is the function of sleep, because it's not really answered yet. And could we sleep shorter and function normally? And actually I will show you a few papers that say yes to this answer. However, if we sleep shorter, we have increased risk of death. So reduced sleep in Drosophila shaker mutants is a first publication that talked about mini-sleep mutation, shaker mutation, and in Drosophila, and one tried to imply something about mammalian sleep. So the mini-sleep mutation, fruit flies slept only third the duration of normal Drosophilas, and that mutation affected ion channels so that plasma membrane was repolarized. And there was a decrease in the duration of sleep episodes, but not the number of sleep episodes. Circadian rhythm was still present. Sleep was more intense. You could say maybe more effective because there were fewer awakenings, and we had increased duration of sleep episodes. And when those interesting Drosophilas were awake, they moved and explored normally. They weren't hyperactive, and they seemed to have normal locomotor activity. So I'm not sure, but I think that if this was in this publication, but those individuals I will talk about, like people that also slept for less every day, had increased risk of death. They died at a younger age. So even though this model seems to be perfect, like you sleep not much, but you behave normally, actually you may die earlier. And shaker mutation, so that mini-sleep mutation is a mutation in shaker gene. And this gene was found to regulate sleep need and efficiency, and is expressed mostly in the brain. Transcripts were found the same in similarly distributed in mini-sleep and wild-type flies in their head. So the mutation is in ion channels, and all non-shaker ion channel mutants tested were normal sleepers. Whereas if the mutation was in the shaker ion channel, then Drosophila slept for shorter, for example, to standard deviations less than the average of both genders. And you see here one mutation, another mutation in Drosophila over many experiments made. And conclusion drawn was that this functional shaker gene appears to lead to a short sleeping phenotype regardless of a particular mutation. And the mutation in that in particular was made in voltage-gated potassium channels. And it produced extreme short sleeping phenotype with preserved performance, as I said. And why the performance was preserved. So sleep, this is like irrelevant because we can take home messages are that sleep can be affected by a single gene mutation. It opens the way to identifying specific mechanisms by which sleep performs its function at the cellular level, and suggests that it may be possible to develop molecular tools that prolong wakefulness and promote short periods of highly restorative sleep. And you see here, this is from paper, still from the same paper. So promoting short periods of highly restorative sleep would mean promoting REM sleep, as I understood. So it's a short period phase, like it's short period phase of the sleep, and during REM sleep, our memories consolidate so that the more that sleep we have, the more it's efficient. So what can we learn from Drosophila? How could we apply the knowledge to mammals? So it may be responsible to develop molecular tools that prolong wakefulness and promote short periods of highly restorative sleep. Shaker locus encodes the alpha subunit of a tetrameric voltage-gated potassium channel that controls membrane repolarization after action potentials and presynaptic transmitter release. And we could generate sleep rhythms by doing mutations and clock genes. Now, the second paper I read was about Marvin Syndrome. I found it very interesting syndrome because even though it's rare disorder, it is marked with sleeplessness and has been associated with autoantibodies against voltage-dependent potassium channels that may have crossed the blood-brain barrier, as was said in the previous paper I was talking about. But without insomnia, the syndrome includes symptoms such as pain, weight loss, hallucinations. And in this paper, a man, 76 years old, was described. So he has absent a circadian rhythm of prolactin and melatonin. And this is thought to lead to central disturbance. And plasma exchange was performed to that person, and symptoms actually improved. He eventually died 11 months later after the plasma exchange. And autopsy of his brain, from his body showed that he died from cancer, but in the brain was found normal thalamus and microinfarct in the hippocampus, sorry. And voltage-gated potassium channels are actually in the periphery too, and antibodies against them produce direct effects that lead to central symptoms. And also, this patient had cardiac arrhythmia. And the third paper is just one slide. So it's about an extreme case of healthy insomnia. It's quite an old paper, but quite interesting. So women claimed to sleep for less than 1 hour each night. And during one observation period, she remained awake for 56 hours, but had only 99 minutes of sleep on the following night. In the second study, all night, electroencephalograms and EOG records were made of her sleep for 5 successive nights. During this week, she averaged 67 minutes sleep per night without any signs of sleep deprivation. So this seems incredible. Maybe we would like to sleep such a period of time, but and this woman does not seem to have increased death rate. But a specific paper really said that these patients die earlier than expected, and this is thought to be due to genetic mutation, and that is being transmitted through from offspring to offspring. Now, penultimate paper is about 2 cases of healthy insomnia. And the paper reported 2 healthy men who normally slept for only about 3 hours per 24 hours. And we spent a large amount of their sleep in orthodox sleep stages 3 and 4, and showed evidence of a high pressure towards REM sleep at the expense of stage 2 sleep. So again, this seemed to be like more efficient sleep that was mentioned in first paper. So maybe if we increased amount of the stage 4 sleep and reduced amount of stage 2 sleep, for example, maybe we could sleep for shorter and be still alert and awake during the day. And last paper is about individual differences in adult human sleep and wakefulness, leitmotif for a research agenda. So short sleepers have reduced levels of stage 2 and rapid eye movement sleep, but similar amounts of slow wave sleep compared to long sleepers, supporting a claim that short and long sleepers do not differ in non-REM sleep homeostasis. So this is kind of oppositely from what we have just heard, because non-REM sleep homeostasis is kind of said to be not changing through various people, indicated that non-REM sleep is actually important, and without that, and it's actually needed. So here it is. Thank you very much for your attention. Here are just a few quotations I found about the sleep. Do you have any questions?
It's incredible. I personally have a question about the. So you mentioned that it's bad to exercise in the evening because of the rising cortisol levels. I think it was in very beginning, but is there a time scale for that? Because like it's a personal question, because I personally exercise in the evening, and I actually find it to increase my quality of sleep, like. But I do it like 3 or 4 hours before going to bed, so I'm not sure if it's.
Maybe when you reduce your cortisol that was accumulated due to intensive focus on schedules and studies.
Yeah, probably.
I think it's important to reduce cortisol in the evenings, like to avoid arguments or stressful emails or something, so that we could fall asleep without cortisol in our blood.
Oh yeah, very much needed modification. Oh, sorry, you muted.
Do you have any other questions, or we can finish? And maybe you want to say now that you want another topic for another journal club?
I think I'm pretty good with the topics you proposed.
Okay.
Yeah, but if any of you guys have any other questions, please fire away.
I think it's fine. I'm not an expert, so I may not be able to answer, complicated questions.
But damn, like your level of preparation with Monte, it's just so astonishing. Like, well, I would personally think that you've written prepared an essay or a dissertation on this topic.
Oh,
There's
no,
so many
far
details.
from that.
You talk about this in a really confident way, so it's amazing.
Oh, really? Thank you. Nice to hear that. Thank you very much. So thank you very much for your attention. I hope you enjoyed it. And if you want to recheck it, you can check it on YouTube, I guess. So thank you very much and goodbye.
Thank you everyone for attending. Have a nice evening.